point? Oh, this
Oh, yeah, it's just here, and that's, I think it just, that's all they need. So there's a laser in the middle.
Laser's in the middle, is it?
You don't have to be looking around.
Oh, no, laser seems to be doing something else. No, that, oh, I see, that's the enter button. This must be it.
Oh, yeah, yeah, sorry.
Yeah.
And that one makes the screen go weird.
Yes, I don't know what I've done.
Otherwise, they basically said, like,
Right, okay. So, I think, well, I think what you're saying is that you're going to have just open mics going just because they want to make recordings. But I guess don't feel obliged to have to talk to the microphone if you don't want to. But I think these mics will live, they just have our audio recording sessions. How do you pronounce your last name? It sounds Corniola.
Yeah.
It's alright. Where are you from? Zurich? It's just nice to be able to pronounce somebody's name properly. Okay, so welcome everybody to today's session. I'm Grant Hopcraft. I'm going to be chairing this session. We've got 7 speakers. We're still waiting for the upload of the 8th speaker, which is Roberto. If anybody knows Roberto, if you ask him maybe to upload his presentation as things go along. For the speakers, we have 15 minutes. We're going to have hopefully 13 minutes roughly of presentation. I have 3 signs here. One I'm going to put up at 3 minutes when you have 3 minutes left. One when you have 1 minute, so 12 minutes, okay, 1 minute left, and then 0. Once they hit 0, you're on to question time, okay? And then if the 15 minutes comes along, we're going to have to move on to the next person, okay? I guess a few comments. obviously, doors, emergency exits are back this way. Toilets, I think everybody knows where those are now. In fact, just out in the foyer out here. In case of a fire alarm, I think we all know what to do. We all leave the building. We have, what else do we need to do? Right, there's, the mics are live, but we're not going to necessarily be speaking into them, but they're just for sound recording. There's plenty of seats over here as well, if people would like. Okay, so I think we'll get started. The first presentation is by Jenny MacDonald, and she's going to be talking about exploring predictors of demographic strategies across life history. So please, Jenny, I'll come up here and I'll load your presentation for you.
Perfect. Do I just click on here?
You can, there's a pointer on this. So speakers, there's a pointer on the button here at the top.
And then it's just
I think this is the forward and back.
yeah.
Okay?
Perfect, thank
Okay.
you. Okay. All right. So hi everyone. So today, I'm talking about demographic strategies, predominantly across plant populations. So how do populations respond to different environmental changes, and is this reflected in other demographic traits or life history traits? So I'm briefly going to talk you through what I mean by demographic strategies, look at this across plant populations and explore potential predictors or correlates of demographic strategies, and then a little bit on what I'm working on now that may explain some of the variation that we observe. So life history, the contribution of demographic rates to population growth varies amongst different life history traits. If you're long-lived, your survival is very important. If you're short-lived and you've got fast generation times, then perhaps reproduction is relatively more important. So demographic rates contribute unevenly to population growth. And then all these populations are subject to environmental changes, so variation in weather, predation events, herbivory pressure, for example. And so when the environment varies, demographic rates have to vary too. Life history constraints prevent organisms from protecting everything. So under these circumstances, it makes sense for populations to protect those rates that are most important to their population growth, and to perhaps suffer variability in those rates that are least important. And this is basically, this process is a demographic buffering process, and the formal kind of definition is that selection should favour genotypes that minimise variance in those demographic rates to which fitness is most sensitive. So throughout this talk, I'm going to, population growth is a proxy for fitness. So this makes sense that organisms are protecting their most important traits by reducing variation, but how do we actually observe that in wild populations? So you can see evidence for demographic buffering when you observe a negative correlation between the importance of a demographic rate, as indicated by the sensitivity of population growth to changes in that rate, and its variation. So I've done this across around just over 100 plant populations, and you see this negative relationship emerge. And this isn't novel, this has been found across lots of different taxa. An alternative strategy, however, that's less widely explored in empirical data but has theoretical underpinnings is lability, whereby populations may track environmental changes, and under some circumstances, actually variation can have a positive effect on fitness. So this implies that there could be a positive correlation could occur. So I'm looking at whether buffering and lability occurs across wild populations. However, to do that, and before I jump into any analysis, I need to account for the different mean variance constraints that are on vital rates. So here we've got simulated data, and this is data on the left is data of plant populations, and we've got rates at the top here. So by rates, I mean things that aren't constrained to lie between 0 and 1, such as reproductive rates. And you'll find that they generally increase, the variance increases with the mean on the x-axis there. And then you've got demographic probabilities, such as your survival and your growth probabilities, and they're obviously bounded between 0 and 1. So as you reach the outer bounds, variance decreases. So there has been some concerns that perhaps what we're seeing is an evolutionary effect, but perhaps those vital rates with the steepest selection gradients, the ones that are most important to your population growth, they're just naturally constrained to have low variance. So, for example, if you've got a survival rate close to 1, you can imagine that could be very beneficial for your fitness. However, just because it's close to 1, it's going to have low variance. So you can see the worry is negative relationships emerge. So I simulated data to reflect the mean variance constraints of plants, and I tested it for demographic strategies. And the majority of simulations found negative correlations. So just if you randomly pick numbers, you're going to find evidence for demographic buffering, which of course is not what we would expect or what we'd want. So that just emphasizes the importance before, when thinking about these things, that we need to account for the underlying mean variance relationships. So with that in mind, I wanted to explore this across plants. So to do this, I used Compadre, which is a freely available online database filled with lots of plant population models. I extracted populations that had been studied over a certain time period, so then I could estimate variability of the demographic rates, and then also the sensitivity, the importance of those rates to population growth. So then I could correlate these. So here are the density plots of the correlation coefficients. So we've got uncorrected, so a naive analysis assuming there's no mean variance problems, and then corrected where I've accounted for the dissimilar demographic scales. So I'm not going to go into the transformation approach because that was my talk last year, but basically you can transform the parameters so they're comparable across estimates and account for those constraints that I showed you on an earlier graph. And what I find is that evidence shifts sort of along the x-axis so that you get more positive correlations. And so a positive implies stability and a negative implies buffering. However, this isn't just a shift over. Actually, within these plots, plant populations are moving about. So some are becoming buffered and some are becoming labile. And then equally, we've got ones in the middle that there's no strong evidence in either direction. So I set out to explore what would be driving this variation we observe, just using simple proxies for different measures. So demographic variability, looking at average standard deviation across population matrices, perhaps environmental variability, so considering ecoregion and latitude, life history traits, so considering life expectancy and the complexity of stage classes, and also invasive status of plants. And this is what I find. So these are the standardized effect sizes and the 95% credible intervals. So not much jumps out. There doesn't seem to be any strong predictors apart from the number of stage classes. So focusing in on them. So this could be real. Perhaps if your plant population with really complex life stages, you're more able to spread the risk and buffer yourself. But it could be mathematical. If you've got a bigger matrix, you've got more demographic parameters, you've got more power more statistical power to detect a signal. And if you're a smaller matrix, if your plant populations are summarized by a smaller matrix, then perhaps the demographic rates have been averaged out and you've lost some of the variation that you would naturally see, so you can't detect any strategy. But with this in mind, accounting for this, I do find strong evidence for phylogeny. It accounted for over 50% of the variation in demographic strategies. And so you can see that there's there's no strong overwhelming evidence anymore for demographic buffering. Some plants are buffered, some are labile, and some lie in between, and we don't find any signal. So this is where I'm at currently, and I'm trying to now tease apart what's driving this different variation instead of just using the kind of proxies that I used in the initial analysis. So to do that, as a disclaimer, very much a work in progress and everything could change, but I'm looking at using ternary plots that whereby previously it's been found that elasticities, the survival, growth, and fecundity can explain variance in different life history traits. In 1993, it was found that where populations lie in this ternary space determined what kind of plant, determined plant type, And then in the noughties, it was found that these can also describe demographic parameters and also life history traits. So I'm using these to see if actually these can explain variation in demographic strategies. So to do that, I plotted the relative elasticities to survival, fecundity, and growth, and I've color-coded them according to demographic strategy, with dark being buffered and light being labile, and trying to explore whether where you lie in this plotting space indicates your strategy. So this gets quite messy because we've got correlated compositional variables, and I'm trying to figure out ways in which I can analyze this effectively. So at the moment, I'm using a compositional principal component analysis, which distills the variation in these three axes into just one uncorrelated measurement. So the first principal component of where these points lie is represented by this curve here, and then I can assign you get a principal component score for every single population, and I can see if where these populations lie is related to demographic strategy. So this was literally last week, and I just popped it in a GLM, and it was significant. This is a bit of a boring graph, but because it's new, I just popped it in. So that's the principal component score on the bottom, and the correlation. This was a significant effect. If I pop back to this graph here, which is a bit more easier to explain, it appears that as elasticity to survival becomes more important, as you're moving down this curved line, plants are becoming more demographically buffered. So there's something there going on that perhaps long-lived species more likely to be buffered, and as you move away from that corner, then you're more likely to have demographic lability. So this is where I'm at. So in summary, not all plants are buffered. I find phylogenetic signals suggesting there's some evolutionary nuances, and using this classical approach to analysis with these previously developed ternary plots suggests that life history might determine a plant's demographic strategy. So thanks for listening, and thanks to everyone there for their help.
Thank you, Jenny. So we've got a few minutes for questions. If anybody has any questions for Jenny.
Please. Hi, Jenny. Thank you so much for that. That was excellent. I was wondering the ternary plots, if you have to take into account the fact that longer-lived things tend to have higher dimensionalities, and if you have been able to correct for that in the decomposition that you showed.
So yeah, no, not at the moment. So I'd really want to be putting these principal components into sort of a more like an MCMC GLM with lots of different effects. At the moment, it was just the ternary plot, but I need to account for the stage classes and the greater complexity. So yes, it could all change, but yes, I would definitely need to account for that.
In the plots that you showed, where you were concerned about the parameters that are constrained between 0 and 1.
Yeah, the hump distribution, yeah.
That's partly a function of choosing to use survival probability as the parameter. Do you know if anybody has tried doing this where you use, for example, the logit transform of survival probability, or the mortality rate, variables that aren't constrained between 0 and 1, and would give you more of a range there? So
that's the transformation approach that I applied, was the logit transformation to stabilize the variance and to stabilize the variance stabilized sensitivity analysis. So yes, if you apply a logit transformation, then that mean variance constraint disappears. So yeah, that's exactly what I've done. But I haven't looked at like survival versus mortality, but used that transformation.
Any other questions? Good. Thank you.
Thank you.
Right, so our next speaker is Tina Cornioli, and I'm going to load up your presentation for you here. So Tina's going to be talking to us about, here we go, body mass of wandering albatross, fathers. Thank you.
Thank you very much for the introduction. It's a pleasure to be talking here today. I'd like to discuss with you the effects of phenotypic traits on life history processes, and in particular the effect of body mass of fathers, but not of mothers wandering albatrosses on life history processes. To understand how population fluctuates, or population dynamics in general, I think it's important to be able to identify the proximate drivers of life history processes. And body mass has been identified in several species of mammals and birds to impact life history processes. The concept is that heavier individuals have more body reserve and can generally perform better, so they have higher survival rates or better reproductive output. Typically, if you have body mass of, whether the body mass of one parent or of both parents will have an effect on the reproductive outcome will depend on the parental care system that the species has. Typically, if you have only one parent that is caring for the offspring, it's the body mass of that parent that should impact reproductive output. And this is the case in the majority of the mammal species. Now, in species providing bi-parental care, you would expect the body mass of both parents to have an effect on reproductive outcome. Now, as we know from our own species, it's not because we provide bi-parental care that both parents perform the same role, invest the same into reproduction, or contribute equally to reproduction. So I'd like to take 1 or 2 minutes to give a definition of these three concepts for the purpose of this talk. And I will take you outside of the field of ecology, but remaining in academia. I would like to introduce you to Anne and John. Anne and John recently obtained their PhD. And while they were PhD students, they both, oh, sorry, you can't see the entire slide. They both did literature search. They both did field work. In essence, they performed the same role. They both invested a lot of time and effort into the PhD. They both worked until very late at night. They both gave up all form of social lives during that time. They had the same investment. However, Anne, during her PhD, managed to contribute 10 articles to the field of ecology, whereas John contributed 4 articles to the field of ecology. So despite performing the same role, having the same investment, there was a different contribution. So now let's go back to ecology. Parental role refers to the tasks that are performed by each parent. Parental investment refers to how much energy does each parent spend for reproduction. And finally, parental contribution is how much output arises from each parent's investment. Now, at the end of the day, what is going to matter for reproductive output is how much energy has been given in total. So it's only parental contribution that should have an effect on the outcome of reproduction. So you would expect the body mass of the parent contributing the most to reproductive output to have an effect. And a great group to investigate this are seabirds, because they provide bi-parental care. They would usually share incubation duty, chick feeding duty, chick rearing duty, with some variance in the way they partition role, invest, or contribute. And I would like to draw your attention to the wandering albatross. The wandering albatross is a bird where parental role are roughly equally partitioned, but the fathers provide more food to the chick than the mothers, and on top of that, a chick has only one mother. On top of that, this is interesting because in 2012, Henri Weimarskirch and his colleagues, they found out that the individuals today are on average 1 kilo heavier than they were 20 years ago. And this has happened in parallel with an increase in wind speed and an increase in breeding success. So that last graph down there is breeding success. And I think it's very tempting when you see those graphs to think that there's a connection among them. And my objective was therefore to investigate the impact of body mass on life history processes in the wandering albatross, a species providing bi-parental care with a specific focus on reproduction. The life history processes that I examined were either related to survival or reproduction. I examined juvenile survival, that is the survival of the individual during their first 2 years. And we related this to chick mass. And we also looked at adult survival, so that is the survival of individuals that have bred at least once in their lifetime, and we related this to adult body mass. And then we looked at 3 different life history processes related to reproduction. The probability to breed, so simply whether an individual has bred in a given year. Breeding success, whether or not an individual has successfully fledged a chick. And finally, chick mass of the offspring. And all of these 3 processes were related to adult body mass. And we performed this for males and females. Now, maybe a few words about the wandering albatross. It's a species that is found in the sub-Antarctic. And the population that I was working on is one from the Crozet Archipelago in the southern Indian Ocean. This is a fantastic database to work with. Data have been collected since 1966, and today almost every single chick that is born on this island is ringed. And that allows us to track the individual life histories. The wandering albatross typically starts breeding around 10 years old. They produce 1 egg and raise 1 chick. And if they successfully manage to fledge a chick, then they usually skip breeding the next year. And on top of having individual life history records, there was 662 adult mass measurements and 412 chick mass measurement that I could use for my analyses. And I used them in a survival analysis and reproduction analysis. I'm sorry about the slide. But what it says is that I performed the survival analysis in a capture-mark-recapture framework using eSearch to account for imperfect detection. And for the reproductive rate, I fitted generalized linear mixed effect model in R with a binomial distribution for breeding probability and breeding success, and with a Gaussian distribution for chick mass. And then in both cases, I performed a model selection based on AIC and parsimony, so simply selecting the model with the lowest AIC and all the models following that that had fewer parameters within 2 delta AICs from the model with the lowest one. A brief summary of results. So what we found, oops, not what I wanted to do. So what we found is that female body mass impacted none of the rates, none of the processes that we examined, and that body mass of males impacted juvenile survival, adult survival, breeding success, and also that there was a relationship between body mass of males and chick mass of sons, but interestingly, no effect of body mass of males on chick mass of daughters. I'll come back to that in a minute. What happened was the survival. So what is on the y-axis of that graph is adult survival, and on the one on the right, juvenile survival. And what you see is that for males, with increasing body mass, survival increases, whereas it's constant for female. And we find this both for adult survival and juvenile survival. This sex-specific effect might be due to the higher metabolism of male, but I'm not entirely sure that this is the case. Now, regarding reproduction, we found no effect of body mass on breeding probability. Now, if I'm honest, I cannot exclude that this is an artifact. Why? Because when they skip breeding, they don't usually come back on the island. But for us to be able to measure their mass, they need to be present on the island. We cannot measure them when they are at sea. So it might be that the few individuals for which we have a body mass that did not breed might simply not have bred because of other reasons unrelated to their body condition. So I put a question mark up there. Next, we find a mass-dependent breeding success in males. So this graph actually shows that breeding success in males is constant until their late 20s. This is age on the bottom. And then it suddenly starts declining. This is nothing new. This has already been published before in the Wandering Albatross. But what I'm showing here is that within one age category, breeding success increases with body mass. So heavier birds within their age class have higher breeding success. And we find no such relationship for females. This makes sense if you remember what I told you at the beginning, that the males is the individual, is the parent contributing the most to reproduction because they bring more food to the chick. We also found that heavier fathers have heavier sons, which is the blue line. The the y-axis is chick body mass. And no matter how heavy the father is, the chick body mass of the daughter doesn't change. And this actually makes sense, because if you remember that graph here, we found that with increasing chick mass, sons have higher juvenile survival, but this rate is constant for female. So investing more in sons improves survival of sons, but investing more in daughters would be wasted because it provides no additional benefit to them. So to wrap up, what I've been discussing so far is the effect of body mass on life history processes, in particular the effect of male body mass on life history processes, but this is by far not the full picture. I mean, it's very likely that body mass are impacted by environmental factors. If you remember this graph here at the bottom, we found that, well, we've seen that the mass of the wandering albatrosses has increased in parallel to wind speed, so it's tempting to think that there is a correlation between these things. And then life history processes are going to have an impact on population dynamics. So the next step would be to be able to identify the arrow going from environmental factor to male body mass and the arrow going from life history processes to population dynamics, and then later be able to construct a global model that would allow us to investigate the effect of environmental factor on population dynamic through the effect of through the effect of traits that impacts fitness. Okay, and that leads me to the end of my talk. I'd like to thank you all for your attention, and of course all the field workers that have been working on Crozet since 1966. Dominique Besson and Karine Delors, who's been managing this chaotic database, and also Koen van Bentham and Marine Després for their helpful discussion. And some funding agency, which also includes, on the bottom right, the IPEV, the Institut Polaire Français, which is financing the research on the Crozet Island. Thank you.
Thank you. So we've got a couple of minutes for questions. Yeah, in the back there.
Hi. How do you account for body size in your analysis? For example, I'm heavier than this guy here, but I'm also quite a lot bigger than him. So are these heavy birds bigger birds or birds in better body condition?
So that is a very fair point. Ideally, it would have been great to have another metric, like tarsus lengths, but there is by far not as many tarsus lengths measurements than body mass measurements. So,
what's your feeling then?
So my feeling is that it's okay. It's from having been in the field, I don't think that would change the conclusion very much, but.
Do you think the birds are bigger or in better condition?
Oh, no, I think they're in better condition. Yeah.
What about the sex-specific reproductive costs? For example, do successful males pay a cost in terms of increased mortality or lower reproductive success the next year?
Okay, so we've also checked for that because we wanted to figure out whether this had a cost. But it turns out that no matter whether they had a son or a daughter the previous year or 2 years previous in time, their reproductive success in the future is not affected. And they also don't produce more daughters or more sons. There's no differences.
Good. I think we're going to have to move on to the next speaker. Thank you very much, Tony.
So hello everyone. So I'll be presenting some of the work I conducted for my master's thesis at the University of Zurich, where I looked at determinants of vital rates in the Siberian jay. So doesn't work anyways. But yeah, so one of the keys to understanding how environmental change will, maybe we can change the format in the.
Yeah, we can change the format.
If you go to the design. Show fit. Great.
Is that okay?
That's perfect.
Take two.
Yeah. So I'll skip this slide. So one of the keys to understanding how environmental change will impact wildlife lies in understanding the relationship between the environment and vital rates. However, the underlying mechanisms of these relationships are often poorly understood, and often the effects are not direct, but instead trait-mediated, so that the environment influences a trait which influences vital rates. For example, food availability may influence body mass, which in turn influences survival, or weather may influence phenology, which in turn influences reproductive success. Of course, the effects can also be direct, such as the effect of predation on survival. And in addition, traits may not only mediate the effect of the environment, but may also modify it, so that individuals with different trait values respond differently to the same environmental conditions. For example, may individuals with a larger body mass have higher survival rates when the weather conditions are harsh. So incorporating traits when studying the effect of the environment on vital rates may help us to gain a more mechanistic understanding of the underlying processes, and may also help us to make better predictions of how environmental change will impact wildlife. So in my project, I investigated the role played by traits in mediating and modifying environmental effects on vital rates in the Siberian jay, which is a non-migratory corvid residing in the boreal forests of the northern Palearctic. So the Siberian jays live in group, each inhabiting a territory, and in every group there's one breeding pair who produce offspring each year. And within each brood there is a dominance hierarchy where the larger offsprings are dominant. And the dominant offspring force their subordinate siblings to disperse and settle in another territory. And these are thus referred to as early disperser prebreeders. And the dominant siblings stay on in their natal territory for up to 4 years and are referred to as delay disperser prebreeders. And the delay disperser prebreeders are dominant over the early disperser prebreeders, and breeders are dominant over both. But once you become a breeder, dispersal history does not matter anymore for social status. But dispersal history is still important throughout an individual's life, as delay dispersers tend to have higher survival rates and reproductive success throughout their lives. So in the Siberian jay, we know that social status is important for survival and that dispersal history is important for reproductive success. However, we know very little about how vital rates are influenced by body mass, which Tina explained is a very important predictor of vital rates in many species. And studying the effect of body mass in species with the social dominance hierarchy may be particularly interesting, since your social status may influence your access to resources, which in turn influences body mass. And in addition, body mass also tends to be quite responsive to the environment. And so because of this, we expected there to be body mass-mediated effects of the environment on vital rates in the Siberian jay. But we also expected direct effects, because we know that in this system, predation is the main predictor of both survival and reproductive success. And we expect this effect, of course, to be direct. And we also know that forest cover mediates the effect of predation, where if you're in a more open forest, you're more easily spotted by predators and then eaten. So, on the other hand, we expected effects of weather to be body mass mediated, as weather may influence both food availability and thermoregulation, both of which are strongly related to body mass. Finally, we also expected traits to interact with the environment so that individuals of different trait values would respond differently to the same environmental conditions. So, for example, may individuals with a higher social status cope better with low food availability, or individuals with a larger body mass may cope better with harsh weather conditions. So to straighten out these question marks, we used data from a long-term study site in northern Sweden, which is in the western part of the distribution of the Siberian jay. And this study site was established in 1989, and at the moment consists of 70 territories in which all individuals are ringed. And some of the territories are located in unmanaged forest and a nature reserve, while others are located in managed forest. And as you can see, forest management has a large impact on forest structure. So in this study area, 2 censuses are carried out per year, so one in March and one in September. And as you can see, the reproductive season and early dispersal falls completely outside of the census period. So during each census, we record who is present in each territory and assess their breeding status. And in addition, for new individuals, we also sex them and classify them as early or delayed dispersers. And upon ringing and during subsequent recaptures, we also weighed individuals. And in March, the fieldwork is carried out on skis, which is very fun. Okay, so we started by looking at the effect of environment and social status on body mass. And we did this using linear mixed models, where we studied the 2 census periods separately, and we compared models using AIC. So we found both environmental and social effects on body mass. So to the left, you can see the effect of proportion of unmanaged forest on autumn body mass. So we found that there was a positive effect, and this could potentially be explained by individuals in the more managed open forest being an easier prey for the predators, and thus they keep their body mass lower as lighter birds can escape predators better. And on the right, you can see the effect of dispersal history and breeding status on body mass in spring. And here you can see that the breeders are heavier than the pre-breeders, and that delayed dispersers are heavier than early dispersers. So it seems as if social status impacts body mass, or individuals of a higher social status have a larger body mass, probably because they have easier access to resources. Following this, we studied determinants of vital rates. So first we looked at apparent survival and studied the effect of environment, social status, and body mass. And this was done in eSearch, so in a mark recapture framework. And the rates were assessed or estimated from census to census, so in 6-month intervals. And we also looked at whether environment interacted with either social status or body mass. And second, we also looked at reproductive success using generalized linear mixed models. And we looked at the effect of body mass and the environment and interactions between the two. And since the census period misses the reproductive season, we used the presence of delayed disperser juveniles at the September census as our response variable. And again, we compared models using AIC. So we found that social status was also important for a parent's survival. So here you can see the effect of dispersal history and breeder status on the parent's survival. And as you can see, breeders have a higher survival rate than pre-breeders. And in summer, there was also a difference between early and delayed dispersers, where in breeders, the delayed dispersers had a higher survival rate, and this is not because of differences in social status, as I explained before, but rather that delayed dispersers tend to obtain higher quality breeding territories. The effect on pre-breeders was opposite, which was unexpected since the delayed dispersers are dominant over the early dispersers, but this could potentially be explained by different emigration rates out of the study system, and this could potentially also explain the difference between breeders and pre-breeders. We also found environmental effects on apparent survival, so we identified a positive effect of snow depth and a positive effect of proportion of unmanaged forest. And the positive effect of snow depth could be because when snow is more abundant, the alternative and preferred prey of the predators of Siberian jays are easier to spot and thus eaten instead. And the positive effect of proportion of unmanaged forest was again probably because predation rates are higher in the managed forest, which is more open. There's also 2 environmental effects on reproductive success. A positive effect of hatching temperatures, so mean daily temperature just before and during hatching. And this may be because when it's colder, the thermoregulatory costs are higher, and so the parents need to make more frequent provisioning trips, which draws attention to the nest and increases predation risk. And the distance to closest house is potentially also explained by predation, where nest predator abundance is higher close to human settlement. So while we expected traits to both mediate and modify the effect of the environment on vital rates, we did not find these relationships. But we did find that both body mass and vital rates were influenced by social status and the environment. And potentially, the reason that body mass did not mediate effects is because predation is the strongest effect in this system, and it tends to be direct. And in addition, for birds, they need to trade off any positive effects of higher body mass against the higher predation risk when you're fatter. And for this reason, they keep their body mass within a more narrow window, and potentially this explains why body mass did not explain more variation. So to conclude, the role played by body mass in the system was very small, and instead the demography was structured by the social system and the environment. And on a larger scale, this suggests that the usefulness of studying traits may depend on the focal species. So for example, studying body mass may be more useful in, for example, mammals than in birds, where any benefits need to be traded off against reduced flight ability. And we also need to account for the environmental effects that are important in this specific system, where here predation was very important, and as such we did not find any trait-mediated effects potentially. So I just want to end with thanking my supervisors and of course also all the field workers throughout the years. And thanks to you all for listening.
Thank you, Higgins. Okay, a few minutes for questions.
Siberian jays, I believe, are caching food.
Mm-hmm.
Is that correct? So do you think that could interact with body mass not being that important? Because if you have research
yes,
elsewhere.
Absolutely. I think it would be very interesting to look at the hordes and see if that could be a trait potentially that explains variation. But we don't really have that kind of data because it's quite difficult to do fieldwork in winter in these conditions. Yeah. Yeah, so this is a bit complicated. We use several different methods. So one is that you can just study the behavior since the breeding pair tolerate their own offspring, whereas they treat the incoming early dispersers aggressively. And then sometimes there's also been some ringing during the breeding season, and then obviously we can know if the individuals stayed in their natal territory if they're dispersed. And finally, we also have some genetic data, so we can see, if they're offspring of the parents or if they're offspring of someone else. Yeah.
Any other question? I was going to ask you one more question,
Mm-hmm.
and that's, it seems like you have a lot of parameters that are related to predation.
Yes.
But are there any direct measures of the number of predators, for instance, in each of these habitats, or the success rate of those predators?
So we have some predator data, which is collected. Like when we do the fieldwork, we also note down predators. So we have this data, but I think the experience of the field workers in identifying predators varies a lot. So it's not. Unfortunately, that would be very interesting. It's such an important effect. So that would be very interesting to see. Yeah.
Good.
Great.
Thanks very much.
Thank you.
So, do you know get it on the big screen from here. There we go. Good. So our next speaker is Cecilka, and she's going to be talking about some sensitivity analysis and the statistical and statistics of lifetime reproductive output. So please.
Right, thank you. First of all, let me start with giving you a quick definition of lifetime reproductive output. It is the number of offspring an individual produces over its entire lifetime. And the mean of lifetime reproductive output can be equivalent to R0, and as such it is an indicator of fitness. However, R0 does not contain any information on variance, and variance in LRO can provide, for example, the opportunity for selection. The aim of my talk, really quickly, is to convince you that besides mean lifetime reproductive output, we should consider variance in lifetime reproductive output, and we have a method to calculate variances from basic demographic data. And what I will present is how to extend this method to incorporate sensitivity analysis. And we have some application of these sensitivity analyses to some example populations, which reveal some interesting patterns. Here I'm showing you that there is actually a lot of variation among individuals in their lifetime reproduction. So we have, for example, does this work? Yes. We have some graphs for humans where the relative frequency of the number of their live-born offspring ranges between 0 and 15 for Norway and the Amish, and you can see that not everybody is having the same number of offspring. Similarly, for kittiwakes, we have the number of chicks that get fledged ranges between 0 and 28. There is a lot of variation. And this variation can actually be caused by different underlying causes. First of all, we can have individual stochasticity, which has two components. First, you have differences in trajectories that individuals follow through their life cycle. Imagine two individuals with a similar mortality scheme. At a given age or stage, one individual may die while another may survive purely by chance. Similarly, for reproduction, you can have stochasticity where individuals of the same age and stage experiencing the same fertility rate might reproduce or they might not. On the other hand, you have actual heterogeneity, actual differences between individuals in some underlying quality. And that would result in individuals experiencing different mortality and fertility schemes. And individual stochasticity, which is basically luck, some individuals might be more lucky than others, that is captured in Markov chains with rewards. And I will quickly show you how to build Markov chains with rewards. We can take any matrix population model, any life cycle described by a matrix population model, and turn it into a Markov chain. We just take all the transitions that happen between ages or stages and put them in the transition matrix U. Then all of the mortalities leading out from those stages is captured with mortality vector M. And once you have died, you are in the absorbing state of death that you can never leave, and you remain there with probability 1. So, and some of you will recognize that the transition matrix U comes from, or is the same transition matrix that is captured in the projection matrix A equals U plus F. But in our case, fertility only comes in to play with the reward matrix, reward being reproduction, and you can get rewarded with different statistical distributions. If you have a chance, if your fertility represents a chance of getting a single offspring, we assume a Bernoulli distribution. But if multiple offspring are produced, then we assume a Poisson distribution. And these distributions help us figure out the statistical moments for our reward matrices. Yes. And we can parameterize all of these using only mortality, fertility, and transition rates. We do this for a set of human populations to illustrate how it works. We have the Netherlands, Sweden, and Japan at 2 points in time. We look at hunter-gatherers, namely the Aceh and the Hadza, and the high fertility population of Hutterites. For these, we only need fertility and mortality. Transitions between age classes are implicit on a yearly basis. And we assume Bernoulli distributed reproduction. On the other hand, I'm showing you a population of trees, Canadian hemlock, where you do need transitions besides fertility and mortality because this is a size classified model. So you can remain in a given size class for a number of years. And we assume Poisson distributed reproduction for this species. Now, I'm skipping ahead quite quickly. What we get from our Markov chains with rewards is mean lifetime reproductive output, which is a function of these two matrices that we get. We get variance in lifetime reproductive output, and from those two we can also calculate Crow's index, the variance over the square of the mean. This is a kind of standardized variance that can provide the opportunity for selection. And this is basically, if all of the variation was heritable, it would be the maximum level of selection possible. So what I am showing you here is mean lifetime reproductive output for all of our human populations. Some are they used to be quite high, now they're quite low, and for our high fertility, hunter-gatherers and Hutterites are doing quite well. And our tree doesn't have a very high mean lifetime reproduction because we're not looking at seed production or seedling production, but at actual recruits to the first size class. Then we can look at Crow's index, and all our human populations are sort of going along, but most noteworthy is that our tree has a lot of opportunity for selection. But these are basically just numbers, and we might wonder how sensitive are these outcomes to changes in the underlying parameters that we've used to parameterize our model. And we can do that also, but I'm not showing you all of the analytical equations. I just want to give you a sense of how it works. So for example, the sensitivity of mean lifetime reproductive output to mortality becomes the following expression. So you have a small change in row 1, mean lifetime reproduction, as a result of a small change in mortality vector mu. It depends on the fundamental matrix and these two matrices that in turn depend on the sensitivity of the transition matrix to mortality rates, and this is actually key, and this is species specific, so you have to figure that out for your species. And from the sensitivity, we can go to elasticity by just taking the sensitivity relative to the actual outcomes and the actual parameters that we get and use. So these are some of the graphs we can produce. These are all of our 9 human populations. On the right, no, that's the left, sorry. We have elasticity of mean, variance, and Crow's index to age-specific mortality. You see that in the case of the mean and the variance, not much is happening. So for on the x-axis we have age. So these are the ages at which the age-specific mortality is perturbed, and there's not much going on. But you see that for the Crow's index, there's actually a high sensitivity to changes in the first 20 years of mortality rate in the first 20 years of life. Similarly, on the right, we have the elasticity of mean, variance, and Crow's index to age-specific fertility. And here you see that, especially in the reproductive ages, an increase in fertility will increase both mean and variance in lifetime reproductive output, but it will actually decrease Crow's index, and how much it will decrease Crow's index really depends on the population. Similarly, for our Canadian hemlock, we now have the exact same graphs, but we have stage on the axis, on the x-axis, and you can identify that some stages are specifically more sensitive to changes in mortality here. For example, the first and the last stage, and both mean and variance, but that, again, Crow's index is not easy to really predict or explain what's going on, because some of these stages are positively sensitive to changing mortality and some are negatively sensitive. And similarly, we have the same kind of story for the elasticities of mean, variance, and Crow's index to age-specific fertility, where especially the last stage, which is the most reproductive stage, with also the highest survival, really shows a positive sensitivity to increasing fertilities. But again, what happens with Crow's index is something that needs some further investigation. So actually, that was almost it already. What I've hoped to have shown you is that we can calculate not just the mean lifetime reproduction, but also the variance in lifetime reproduction. And we can also do sensitivity and elasticity analysis on the outcomes of these methods. And we can do this for a very broad range of life histories. We've shown you some human populations in trees, but basically anything with a matrix projection models can fit in our framework. I've shown you some elasticities of Crow's index, which I think is a very interesting thing to consider, where, for example, the changes in parameters, the elasticity of Crow's index to changes in parameter can be great, even if the elasticities of mean and variances are small, which is something that might merit further exploration. However, there's a big cautionary note on interpretation here, because, like I mentioned before, all of the variation that arises from our model is stochastic. It's based only on probabilities. So none of it is heritable. So talking about the opportunity for selection without any heritable variation is, well, that's kind of strange. So this method really produces a baseline level of variation, and I would urge you all to think about where the variation that you see, that you observe, is actually coming from, because a lot of it could be stochastic. And with that, I would like to thank all the people on this slide and you for listening, and I'm happy to answer any questions that you have.
Great, thanks very much, Silke. We've got 2 minutes for questions, if anybody has any questions for Silke. So,
Yes.
sorry, was there a question? Yeah, go ahead.
The distributions that you use, the Bernini and the Poisson distribution, presumably you have to choose parameters for those that would the actual shape of those Poisson distribution or whatever. How sensitive is the model to that decision?
So in the case of the Poisson distribution, you assume that that the fertility is giving the mean of your Poisson distribution, and from that follows that the variance is also equal to the mean. And we do see that translates down to the results in some ways. I don't have that figure, but it's an interesting thing that you see that could also warrant some further exploration. Yes.
I have a moment to follow up on that. It's an interesting phenomenon that many people collect individual reproductive output data, which means they have both the mean and the variance and all the other distributions of it. They only report the means, because that's what goes into a matrix population model, is the mean fertility of each age or stage in the population. If the variances and distributions were reported, then you could use those directly and you wouldn't have to assume a particular
Yeah, that's what I
form.
was thinking, because it could take a negative binomial form or something like that.
And if
And if you were able to parameterize that from the real data,
that would be ideal, but there, up until now, there hasn't been much motivation for people to actually report that level of detail, but I encourage people to do so, because then you can, in fact, explore different empirical distributions,
it would
which
be
would
interesting
be
to see
really
how
cool.
that affects the
Exactly,
variations of selection.
exactly.
Good. Thanks very much. Thanks again, Sylvia. So we have a quick transition here. We just need to load up a presentation very quickly. Unless it has been loaded already, I don't think so. Is there an extra port there? Can you see that? There we go. That's it there.
Yeah.
Just drag it across there.
I suppose you just click on that. Yep,
Good.
it's all good. Thank you.
Yeah. So please, all yours.
Thank you everybody for coming. My name is Rob Salguero Gomez, and I recently started an ERIC position at the University of Sheffield. The things that I want to talk about today are the effects that clonality may have on the evolution or the escape from senescence, particularly in plants, but what I'm going to be talking about may also affect animals. So the two commonalities in the three pictures that you have got there is that you're looking at plants that live long and are highly clonal. Both seagrasses, Lariat tridentata or Crassiote bush, and the quake and aspen are known for achieving very long lifespans. And the question that I'm trying to address here is whether, as it has been hypothesized before, clonality may be correlated positively with that high longevity and possibly with a low rate of senescence. So before going into further detail, I need to explain what senescence is. From a demographic perspective, it is a decline in the potential for fertility as individuals get older, and also an increase in the risk of mortality also with age. And this is something that, no pun intended, it's been actually one of the oldest research questions asked by mankind that, as far as we know, goes back to Aristotle. Now, senescence has been recently a rather controversial topic. It's been disproven that senescence is universal. That is, that this pattern that you find whereby fertility should go down and the risk of mortality should go up is a demographic signature of all species. And there's been various hypotheses and theories put forward to try to understand why senescence should be universal or not. And the one that I want to focus my attention on, and therefore yours as well, is mutation accumulation, which states that individuals get more decrepit with age simply because our genetic machinery is not perfect. If you get to live one more year, congratulations, but your machinery is not perfect to get rid of the negative mutations that might have come as a byproduct of that. I mentioned before that senescence has been recently shown not to be universal. Senescence seems to be the rule for some species, as you can find here. Both humans and guppies seem to have paid attention to the fundamental works of Williams and Hamilton and other folks. They have got a fertility that declines after first reproduction. They have got a mortality risk that increases after individuals get older. But there's a lot of other species, indeterminate growers, clonal growers, that don't follow that pattern. Namely, you've got some species on your right-hand side for which the fertility potential increases with age, and other ones for which mortality actually declines with age. In other words, the older they get, the better. This poses the following question. That first of all, we stop asking how come that humans senesce, and that we instead start asking what do those species have got in common, and how can we get to the mechanism or mechanisms that may allow for the escape from senescence. And in my lab, we're taking two different approaches. I'm only going to be talking about the comparative one. The other one has got to do with actual fieldwork that we're carrying out in the UK mostly. The two databases that we're using contain matrix models. Jenny did a bit of an overview of what this information is containing there. Namely, people go to the field, collect rates of survival growth, reproduction, recruitment, and clonality when it happens, in blue up there. And it gets published. We then, at the Max Planck and other institutions, collect the information and make it available. The beauty, one of many, that is, of these matrix models is that you can derive a lot of population-level metrics, but also through a lot of methods that have been headed by Hal Caswell and other folks, we can derive life history traits. So we can now derive estimates of mean and variance of life expectancy, as you heard in the previous talk. We can also talk about the rates of senescence. We can calculate the mean and the variance at the age of maturity, et cetera, et cetera. We can also look at the trends of clonality. And these matrix models have become so popular in ecology and evolution that there's heaps of them. They come in different flavors, and they have been following a book example of an exponential increase in publication. What you have got on the left-hand side is the publication records for plants that have used population matrix models since the first introduction of matrix models to ecologists in 1941 up to last week, and the same thing for animals. So there's a lot of this information, and this information is globally replicated. And what this allows us to do is to ask what are the commonalities of these species across different biomes, how do their life history strategies maybe shaping the rates of senescence, and how does the ecoregion where they live affect these outputs. So my colleague Owen Jones in the back of the room and myself have been looking at this question together with Hal Caswell and other folks at the Max Planck. And what we have come to find so far is that there's not such a thing as a strong taxonomic affiliation. That is, animals are now more likely to senesce than plants. And this can be shown in the inserts, where we're looking at Kifitz's entropy. I'll tell you what that is in a second. This can be thought of as an approximation to whether species will undergo senescence if lower than 1, or escape from senescence if greater than 1. What we find here is that there are some taxonomic groups that don't get limited to animals versus plants, for which likelihood of senescence is quite high. And there are other groups, namely some corals and other plants, for which there are some representative taxa that manage to escape from senescence. In recent publications, we have come up with a quantitative framework to try to classify the richness of life history strategies. This work has so far been published in plants, but we're now working with Kevin Healy and Ivan Buckley on extending this to animals as well. And the patterns that I'm going to show you next are pretty much universal also across animals and plants. So what we find is that there's a main axis of variation that tells you where species allocate between growth on the left-hand side and survival on the right-hand side. And we also have identified a second axis of variation that explains a relatively high variation in the life history strategies, going from species that are semelparous, they only reproduce once, on the bottom, and species that are iteroparous, and they reproduce for a long time on the top. The one thing that was missing in this framework so far, in order to understand whether clonality causes individuals and species to be able to escape from senescence, is to actually bring clonality into the mix. So in order to do that, we have had to be a bit careful with the way that matrix models report clonality. As you may know, demography can be carried out at different levels, at the ramlet level or at the genet level, and the comparisons between those two can be quite tricky. There may be some population matrix models for which the demography has been carried out at the genet level, but the species can produce ramets, can produce clones. And so in order to be able to assess exactly what a clonally reproducing species is, we've gone out between Compadre and asked the open access database CLOPA3 whether a species in Compadre, for which we suspect there may be some clonality going on but was not reported, is actually clonal. So after we have gotten rid of those type 1 errors in the Compadre database, we've ended up with 560 plant species. I've then classified them according to whether they only reproduce sexually, whether they reproduce only clonally, or whether they are able to reproduce both sexually and clonally. And the questions that I want to address here are whether the ramettes of clonally only reproducing species can really undergo lower rates of senescence, as it has been hypothesized by Finch and many other funders of aging research. And also whether trade-offs between allocations to sexual reproduction and clonal reproduction may be shaping the outputs on the evolution of senescence or escape from senescence. So I mentioned before kind of like a quantitative word called Kifitz's entropy. I'm just going to jump through this very quickly. What we have done is, using Hal Caswell's approach, we've been able to derive the age-specific curves for survival, for clonality, and for sexual reproduction. And we've used this matrix, this measure of entropy, to see whether a species undergo senescence, they don't undergo senescence, or undergo what's called negative senescence. That is, the rate of mortality actually decline as individuals get older. We've thrown different life history traits as derived from these matrix models into phylogenetically corrected PCA, and the same patterns that I showed before have remained. You've got a fast-low continuum, but also we can now identify a correlation between investments on clonality and growth, and these are orthogonal from investments on sexual reproduction. You can see where the species lie depending on their reproductive strategies. What you're seeing here is the values of Kifitz's entropy. So the darker the color, the higher the rate of senescence. The oranger the color, the more likely to escape from senescence. What this framework shows is that the location on the first axis, the fast-low, and the second axis, the reproductive strategies continuum, can actually tell you about whether the species escapes senescence or not. I've then taken each of the life history traits and grouped them according to whether they reproduce only sexually in pink, clonally in blue, or both in yellow. And what I have been able to find is that species that reproduce only ramettes allocate more to their survival, they grow faster, and they reproduce clonally for longer than any of the other two types of strategies. What's interesting is that, very much in contrary to the expectation, the rates of senescence for clonally reproducing species are actually higher. Kifitz's entropy is lower than 1 than those for which they only reproduce sexually, or both sexually and clonally. So the take-home message of this talk should be that it is only at the ramette level that we can talk. The ramettes senesce faster than the genet sexually reproducing individuals, and that this is most likely driven through a trade-off between how fast you live and how much you allocate to clonality. What I'm working on at the moment is simulating models whereby I can build ramette within genet matrix models and ask whether there's an optimized rate of replacement within those ramettes that would derive or result into different rates of senescence. If you're interested in this question, there's a book coming out next month or the month after. And on that note, I'd like to thank all the people who collected demographic information at the Max Planck and other centers, and my sources of funding. Thank you very much.
Thanks very much, Roberto. Questions?
Hi, Alan.
I was wondering if senescence might actually still possibly be universal, but that for some organisms just don't live long enough to be able to get out that far. That even if eventually the probability, or if you're observing the probability of mortality going down, still after 80 years, it's unlikely to survive further out, to get to a point where then mortality might come back up again. So I'm just wondering if it's sort of like it's rate censored, if you will.
So there's a bit of technical things that we had to do these models. So I have got access internally to over 3,000 animals and plant species. I've only shown you results for about 500. You might question yourself why. Well, because we've been really picky at choosing the models for which we got rid of mortality plateaus. I think this gets to your question. So we've only chosen models for which 99% of the individuals in the population will be dead before mortality plateaus were achieved. 99% of the individuals in the population. Yeah, we're not accounting for that 1% of individuals in the cohort, but the vast majority of the population is either showing senescence or not. This is the shortcut that we're taking. The long-term investment really is to commit myself and my lab my labs into the future to follow in long-term demography, which is something that some people in the room, namely Pedro Quintana Asensio, are very cleverly doing, but it's currently one of the shortcuts, or sorry, the limitations in plant demography.
Good. Thanks very much.
Thank you. There you go. I'll just take this.
Yeah, just pop that.
Thank you.
Right. So. Oh. Full screen. Good. So last speaker for this morning's session is Greg Roth, who's going to be talking about occupancy in time and neglected population variable. Thanks very much.
Hello, everybody. So today I will talk about occupancy time, and I will show you why it's a neglected population variable. And I will start by my main message, or my main result, which is to show you that there exists a set of easy-to-use formulae to calculate the occupancy time from a demographic model. So I will start to explain what are those occupancy times. So any individual, when he goes through his life cycle, is going through a sequence of events. So growing, aging, moving, and so on. So here I give you an example of a life cycle of a seabird, the southern fulmar, that is represented by his reproductive stages. So pre-breeder here, and after he will attempt breeding, being successful or fail, and he can skip breeding. So from such a life cycle, you can follow one individual. Here, if we follow bird 1, from birth to death, and we can record in which state he is at each time step. And if I take another bird, he will have another sequence. So this one would die after five years. So from this sequence of states, it is common in population ecology to incorporate the transition between those states in demographic analysis. And the most common demographic analysis that we can have are the population growth rate, the reproductive rate, or the expected longevity. But none of those analyses describe the time spent in those states. And this is what is the occupancy time. So if we come back to the example here, the occupancy time in the state successful breeder is the number, is the time spent in this state over the individual's life. So here for bird 1 would be bigger than 2, because we don't know his future, but for bird 2, it's 0, because it dies before. So those occupancy time can describe a wide range of demographic measure. So the lifetime breeding success, like in my example, but it could be the time spent in a specific location, if you describe your individual by location, lifetime risk exposure, time spent in a specific health status, and so on. So what is important to realize here is that those occupancy time are function of those pathway, the pathway from birth to death. And those pathways vary between individuals. So as a consequence, the occupancy time vary between individuals. And here I just give you an example of the distribution of the lifetime reproductive success of a population of female deer. Since they have only one offspring per reproduction events, you can associate that to an occupancy time in a state that would be successful reproduction. So you can see here that the distribution, there is a lot of variation between individuals. So since there is variation in this occupancy time, so what do we calculate? Should we focus on the mean, or should we go further and calculate the variance, skewness, or any statistic you want, and in more detail, the probability distribution? And as we saw in the talk of Silka, the variance is actually a very important statistic to calculate for those kind of measures. So how do we calculate those statistics? I will give you a framework here that allows you to calculate those statistics for any occupancy time in a very easy way. So the framework is a framework of absorbing Markov chain, which is the same framework that Silka were talking about. So what do you need to define an absorbing Markov chain? You need a set of living states. So it could be developmental stages, reproductive stages, as in my example, or whatever you want. Usually is the stage that you use to describe the life cycle of the individual. And then you need only the probability transition among those states. So as I said, those absorbing Markov chain are usually already embedded in a life cycle graph. So here, if you go back to the life cycle graph of the southern fulmar, what you do is that you remove the arrow for reproduction, because you only want to model the pathway from birth to death, and you add death, which is the endpoint for each individual. And now what you need to do is to have the probability transition that you associate to each of those arrows. And those probability transition are collected in this matrix U that we call the transition matrix, and M, the mortality probability. So this matrix U is actually already embedded in a projection matrix in a population model. So here, if I give you this simple example of a Leslie matrix with 3 H classes. So a Leslie matrix has this shape here that you can decompose in survival probability here and reproduction rate here. So the U matrix that I'm talking about is this part here. So now you want to compute the occupancy time from this matrix. So let's call a new ij the occupancy time in state I, starting from state j. So what you can do is to compute the fundamental matrix of this absorbing Markov chain, which is a very simple calculation. It's just the identity matrix minus u, and you inverse this matrix. And what you get is an omega by omega matrix that each entry corresponds to the mean occupancy time in a specific state. Usually we are interested in the first column because the first state is the state where a newborn individual would be. And then if you go down the column, you have the mean occupancy time in each state. We'll go back to that in an example later. So the main result here is that from a projection matrix I, you can extract U, compute N. This is very simple. And from there, you have access of a series of statistics on your occupancy time. So the mean is N. The variance is just a little bit more complicated here, but it's just an algebraic calculation with this matrix. So you can do that in one line of code in MATLAB, for example. But you also have access to the probability distribution. So you really have access to any statistic. And one more statistic that I want to point out here is the covariance between the occupancy time and the longevity. So you can also compute the covariance, for example, of the number of reproductive success events and the longevity of an individual. So, and there is a long list of other statistics that I'm not showing you here. So we'll go back to finish to the example of the southern fulmar. So, yeah, I forgot to say that this life cycle is extracted from this paper of January and al., where they describe the life cycle of this bird as it is here. And in this paper, they consider 3 different ice conditions. So those birds are seabirds that are breeding in the coast of Antarctica, and their vital rates are dependent of ice condition. And here you have a favorable ice condition, ordinary ice condition, unfavorable ice condition. And those matrix U, I just took them from this paper. So I didn't do anything, I just extracted from their projection matrix. And now I want to calculate the occupancy time in the state successful breeder. So if you think about that, what it is the lifetime number of successful breeding event. of an individual. Now I can compute the fundamental matrix, which is just identity minus U inverse. And I got this matrix here for the favorable ice condition. So how do you read this matrix? So this is the mean occupancy time matrix. So if you take the first column, which is a column associated to a newborn, which is born in a pre-breeder state, then you will spend 5.8 years in a pre-breeder state on average, and 5.3 years in a successful breeding state. So it means that the lifetime number of successful breeding events, it's on average 5.3. If you sum up here the numbers on a column, what you get is the mean longevity, because they spend time in each state. If you sum all this time, it's just the longevity. So just with this matrix, very simply calculated, you get the mean longevity and the mean lifetime number of successful breeding. So here I show you this for the 3 different ice conditions. So what you can see is that the longevity, the mean longevity, is not at all affected by the ice condition, but the mean lifetime number of successful breeding is highly affected by the ice condition. They almost never successfully breed when it's unfavorable condition. But if you just look at the mean, then you would just look at that, a huge decrease of the lifetime successful breeding. But if you look now at the variance, so I'm showing you the coefficient of variation, which is just standard deviation divided by the mean. So the longevity, again, is not affected by the ice condition. But the lifetime number of successful breeding, you can see that even though the mean decreased a lot, the variation increased a lot as well. So you have a huge variation on an unfavorable condition. And this is due to the fact that they most of the individual doesn't make it to a breeding attempt. But if they make it, then they will be successful more than once time. So there is a huge gap between making to the first attempt or not. And you can see that here, if you look at the correlation between the longevity and the lifetime breeding success, so you can see that for a favorable condition, it's a correlation of almost 1. So they are very related between longevity and lifetime breeding success. But in an unfavorable, this correlation drops, because you can live a long time, since you will almost never make it to the breeding attempt. It doesn't affect your number of success breeding. It's going to be 0 most of the time. So to conclude, I want to tell you that the occupancy time can represent a wide range of demographic measures. And it's not only important to compute their mean, but the variance and probability distribution is also very important. And you can compute all that from a demographic model in a very easy way. And thank you for your attention.
Great, thanks very much. So we have a couple minutes for questions. Anybody have questions? Hunger at the door. Yeah.
What are the other columns in your matrix where you were giving us the length of time in each stage?
This matrix.
Yeah. So what are the second, third?
So here is the starting state. So if you look at the second column, it would be to look at an individual that starts in a successful breeding, which would be really useful if you're interested in adults. So this is, for example, the number for type of adult.
So I've got sort of a related question. This matrix is essentially built from the previous matrices, right?
Yeah, it's just...
So those previous matrices, you're essentially counting the number of individuals in each of those states. Is that right?
No, it comes from the U matrix. And the U matrix are the matrix with the priority transition between stages.
Between states, oh, I see, okay.
Yeah. It's the matrix
so
that is...
based on the probability matrix of transitions.
Yeah,
right.
which you can extract from a projection matrix A, so that you would have in a population matrix model.
Right, so we're coming up for lunch, I think, which is, I think, starting now. And the next set of presentations starts at 3:15, so I think we've got about an hour and a half, something like that, 2 hours until the next session. So please enjoy your lunch. And a big hand of applause to all the speakers and to also the assistants.